Welcome to Provitalks, where nature and science blend to inspire you. Discover the latest in technology, sustainability, and trends in the cosmetics world, thanks to Provital. Let's care together for our industry's commitment to the people and the planet. Provital. Do care.
Welcome to Provitalks. During today's edition, we are going to give more details about the innovation in research techniques and in vitro efficacy studies in the cosmetic world. For this, we once again count with the presence of our expert, Dr. Miguel Asso, with whom we previously discussed some of the most cutting-edge techniques in cosmetics research, specifically artificial skin models, as well as the use of bioinformatic analysis tools. Miguel, welcome back.
Hello. Thank you for having me again in the Provitalks. I am delighted to speak with you.
Thanks to you. Thank you very much for being here again. We saw in our previous edition that if we talk about bioinformatics, big data, or big science, we are talking about huge amounts of data. How are these data transformed into useful conclusions that better inform us about the efficacy of an ingredient or a cosmetic product?
As I told you in our previous conversation, the impact of big data on dermocosmetic science is tremendous. But the complexity of big data analysis presents a new challenge. To give an example that those who listen to us will know, for the study of gene expression, the real-time or quantitative PCR technique is normally used. Before, about 10 years ago or more, conventional or qualitative PCR was used. The step from qualitative to quantitative PCR was a revolution in its day. Since the experiment took much less time, we went from weeks to hours, and we could also analyze many more genes in a single experiment, up to around 88 genes. Well, today, we can already use massive sequencing techniques. Probably the best known technique is RNA-seq, from the English RNA sequencing, which is the most used technique for the study of the transcriptome. Here, let me give you a few numbers. When the sequencing experiment is finished, the machine gives you a simple text file with base sequences, with the letters G, A, T, A, C, A, etc. The only thing is that you get about 26 million of these sequences in fragments of 100 letters. Well, with that, and through a programming language that allows us to implement statistical algorithms, we take the human genome with the sequences of each gene already described and compare them with the sequences of our experiment to know which genes we have detected and in which amount. Typically, after the bioinformatic analysis, we end up with about 12,000 genes, which essentially means that we have measured the total of the genes that are being expressed at that moment. In other words, the transcriptome. Remember, using quantitative PCR, we arrived at about 88 genes.
Yeah, the difference is huge.
In the end, all this is to explain to you how we do our transcriptomic studies. As I have just told you, with these new massive sequencing techniques, we are able to study all the RNA molecules present at a given moment. You know that the set of genes, which is constant in all the cells of the body at all times, is called the genome, but the subset of genes that are being expressed in a specific cell at a specific time is known as the transcriptome. In other words, with transcriptomics, we are taking a photo of a specific place at a specific time.
And from what you tell us, it seems that the study of transcriptomics requires both very advanced machinery and highly specialized knowledge. I understand that there are research groups dedicated exclusively to bioinformatics. Is that true? And these massive sequencing machines, I suppose they will have a high cost. Is that so? And is it worth the investment to acquire this equipment? Is it possible that private companies also can benefit from these structures then?
Indeed, to be able to sequence samples, we need highly specialized equipment at a very high cost. In fact, and for this same reason, this equipment, unlike others such as the thermal cyclers used for PCR, is not usually available in every laboratory due to its high cost and also because it quickly becomes obsolete, because this is a field with a very high innovation rate. Mm-hmm. What is the best equipment today will be obsolete in 3-4 years. And for a much lower cost, you will be able to perform longer and higher quality sequencing. This is why each laboratory extracts its RNA samples and sends them to specialized sequencing companies that centralize these processes. In addition, as you say, a very specific training in bioinformatics programming is needed to later analyze the data. For these two reasons, we thought of participating in the Corbel initiative. Corbel is part of the Horizon program of the European Union. Through Corbel, we were able to initiate a collaboration both with Massive Sequencing Center at the Academy of Science in Athens and with one of the greatest experts in bioinformatics analysis in Europe, the bioinformatics platform of the Max Delbrück Center in Berlin. Mm-hmm. We collaborate with this German bioinformatics platform both to receive technical advice and also to acquire and develop internal capacities at Provital, because it is always beneficial to know yourself the necessary tools to carry out your own analysis. I compare it to a standard collaboration with a CRO. We have all received results in Excel format of an assay carried out with an external company, but possibly we have never stopped at the conclusions that the CRO sends us, but rather we use the Excel program to further analyze the data in our own way. Well, at the massive sequencing level, exactly the same thing happens, but instead of using Excel, we use different programming languages. At Provital, we mainly use the language R, which is the one I prefer, but you also need to know some Unix and Python. Changing the subject, Clara, in relation to your last question, I would like to tell you about a curiosity regarding the Corbel initiative.
Yeah, of course. Please tell us.
Well, Corbel was born by and for the academy, and unfortunately, communication between the academy and the private sector is not always fluid. In fact, I attended the Corbel program coordination meeting, and those responsible for Corbel were very interested in our case because few companies have been able to approach this program. Probably, in my opinion, because the access routes are typical of academic structures, such as research centers and universities, with which companies are not familiar. To our surprise, the Corbel Outreach Department took an interest in our case and used our example to promote interaction between research centers and companies. And we are very proud to have been pioneers in this interconnection between academy and business.
Well, how not to be proud? From what you explained, this interconnection is vital to realizing and really capitalizing on the most advanced technology. And, well, going back to the technical differences between transcriptomics and PCR studies that you are telling us about, from what I understand, the difference lies in the number of genes analyzed, right? In other words, if with PCR we mediate the expression of dozens of genes, with transcriptomics we are talking about tens of thousands. But I would like you to help me understand, what does that mean on a practical level? What advantage will a product whose efficacy has been studied using this technique? What effect does it have, for example, to better understand the mechanism of action of a new product?
Thank you, Clara, for asking these questions, because truth is that from the laboratory, we scientists often get lost in the technical details and move away. We take our feet off the ground and forget to convey the importance of scientific advances. I will try to answer each of your questions to the best of my ability. First, you are right. The difference in practical terms between the PCR that we all know and massive RNA sequencing, transcriptomics, is the number of genes. We went from tens of genes analyzed to tens of thousands. Okay, but it's not just that we measure many more genes, it's that we measure all of them. And this has important implications. One of the consequences that has the greatest impact is that we have a picture of everything that is happening in the cell. Well, everything that is happening to the cell and everything that the cell is doing. If you allow me, I will give you an example that I love about a new product that we are about to launch right now. By the time our listeners hear this podcast, it will probably have been released.
I think I know what you mean, Miguel. Our new launch in body sculpture, right? Obtained from the blackberry, Sculpup. What a great product. How was it possible to see this magnifying effect of shapes in a raw material that has been used so much in cosmetics, but usually for purposes so different from ours?
Before the transcriptomics study, naturally, thanks to classical in vitro efficacy studies, we discovered that the blackberry promoted lipid synthesis and reduced lipolysis in adipocyte cell cultures. This in itself had not been described before and has a high value, so it is patent pending right now. But nevertheless, we wanted to go further. Then it is when we used massive sequencing for the study of the transcriptome. As expected, based on previous experiments, we observed a huge set of genes related to lipid synthesis and degradation, which not only confirms the mechanism of action of the active ingredient, but also makes it more robust. But not only that, the sequencing also revealed many other genes which were not included in our initial hypothesis. That is, genes about which we had not asked anything. But since we measured the expression of all of them, we observed that there were mechanisms of action of the product that turned out to be important, but were unexpected. Among these unexpected mechanisms, we observed that although blackberry-induced collagen synthesis in the dermis, as we saw in previous experiments in the dermal compartment, on the contrary, in the subcutaneous adipose tissue, the active ingredient reduced the expression of the extracellular matrix genes. We did not expect this result in any scenario.
Wow.
Then, when contrasting this surprising data with the scientific literature, we had a memorable eureka moment, which is that it is well described that the process of decompensated obesity with the development of cellulite with orange peel appearance are accompanied by fibrosis in the subcutaneous layer. That is, with an excess of collagen and extracellular matrix proteins where adipocytes grow. So, we were able to conclude that Sculpup not only promotes a volumizing effect, but also does so in a controlled way, avoiding the formation of cellulite. And this is a good example of where transcriptomics has allowed us to go. But there is a long etc. of more mechanisms.
Well, this is truly amazing, Miguel. Thank you very much for sharing this information and also your professional and even personal opinion with us. We really greatly appreciate your time. But honestly, at least I'm left wanting more. Let's see if we can talk again soon about other techniques, because this is really super enriching. We hope to have provided valuable information for our listeners too, with these 2 Provitals dedicated specifically to the innovation and current situation of in vitro research in cosmetics. Thank you very much, everyone, and see you next time.
Thank you very much, Clara. It's been a real pleasure to talk to you.
Bye.
Thank you for listening to this edition of Provitalks. We hope you got some source of inspiration to create and spread your own kind of beauty. For further information, subscribe to our podcast or visit our website at weareprovital.com. Provital. Do care.